All right. I think it's a time, and let's just get started. So, hello everyone. Welcome to the 26th session of the Global Ocean Oxygen Network webinar series. And I'm Professor Minhan from Xiamen University, China. I will be moderating today's webinar. And the Global Ocean Oxygen Network Secretariat, and I'm very happy to welcome you all and thank you all very much to join us. And so before we get started, we would like to bring your attention to this Ocean Oxygen website, where you can find links to past webinars and to good news. I hope this is the website. You can find a lot of information and also good. Good stands for the Global Ocean Oxygen Decade. That's a UN decade endorsed program. And also the Ocean Deoxygenation newsletter that's shown on the right corner of this. And you can also use the QR codes on the screen that will take you right there. Today we have two great speakers. One is Yawei Shen from Xiamen University, and Folco Giomi and from Italy. I think, so again, thank you all very much to join us, and we'll hand over to Yawei Shen, the first speaker, to give her presentation. Yawei, floor is yours.
Yeah, thank you. I'll share my screen.
Sure.
So can you see my screen?
Yeah, perfect.
Yeah, so I'll start my topic. Hello everyone. My name is Yawei Shen. I'm now a postdoc in Xiamen University, China, and it's really a great honor to give a presentation at the GO2NE webinar, and thank you for listening. Sorry because I changed the talk title to this. The oxygenation in marine culture and some biological strategies. First, I would like to discuss the real-world implications of the oxygenation on marine culture. Next, I'm going to talk about what we might be able to do with it from a biological perspective, such as shaping the ability of maricultural animals to tolerate hypoxia. When it comes to biological mechanisms, I'll make it as simple as possible. Though I think most of people here know that since the middle 20th century, the dissolved oxygen concentrations in the open oceans and the coastal waters will continuously decline. It is caused by global warming and the excessive discharge of eutrophic sewage, and has been one of the most important changes occurring in the ocean. We know that hypoxia events negatively impact the survival, development, and reproduction of marine organisms. So because I am studying marine culture animals, so I pay more attention to information on marine culture. One day I put together this map of global hypoxic areas and this map of global maricultural areas. I found that a large fraction of them overlap. It is indicating that hypoxia poses a global hazard to mariculture. So why is that? I'm thinking of this question. We know that the primary cause of the hypoxia in coastal regions is an excessive of nitrogen discharges from rivers. Oxygen is then consumed by the algal blooms and microbial decomposition of organic matter. However, in mariculture areas, there are few special triggers that increase hypoxia events. And because the marine culture system is semi-controlled, not totally controlled, it's very complex with various abiotic and biotic factors uncontrolled. For example, the persistent high temperatures in summer reduce the oxygen solubility in the seawater and increase the oxygen consumption of mass mariculture animals, and high density mariculture and biofouling organisms further impede water circulation and replenishment of dissolved oxygen. Over the past decades, the importance of mariculture has been increasingly emphasized as it supplements declining fisheries and provides an abundance of high-quality animal protein. However, in the past few years, it has been frequently reported that large amounts of mariculture animals died due to serious hypoxia events, including fish, shrimp, crab, oyster, and scallop. It has caused a huge economic loss and jeopardized the sustainable development of aquatic food systems globally. For example, in southern China, around 30% to 60% of abalone die from hypoxia and heat stresses, resulting in losses of $1.4 billion per year. So to mitigate the hypoxia problem, multiple actions might help. As the author of this review articles said, we can reduce anthropogenic nutrient imports from land, reduce greenhouse gas emissions, and reduce waste production. On the other hand, we can take some adaptive measures to reduce stress to marine organisms and fisheries, including build protected areas that can serve as refugia during hypoxia events, and also some other conservation measures in fisheries. But for mariculture, there may be other useful strategies based on biology. These biological strategies aim to enhance the hypoxia tolerance of organisms so that more of them can survive from hypoxia episodes. One approach is One approach is genetic selection. It involves selective breeding to produce organism strains and breeds that are tolerant of hypoxia. Simply put, hypoxia stress will eliminate those who are not able to withstand low oxygen levels. Those that can tolerate hypoxia will be retained as parents in order to raise the following generations. So according to this Mendel's hybridization system, you know that. So according to this serial, these hypoxia tolerant individuals have genotypes that aid in their ability to withstand hypoxia. So after a few generations, we might eventually get a population that can tolerate hypoxia. So another approach is plasticity induction. So what is plasticity? It is not believed that the phenotype of an organism is controlled by genotype and altered by the environment during its development. That's to say, life and environmental history can influence the future responses of an organism. For example, and identical twin sisters who grew up in different environments may look alike, despite they have the same genetic composition. Thus, we wondered whether the stress tolerance of mariculture animals could be altered based on plasticity induction, as shown in this figure. Some environmental stimulus is applied at a certain life stage, and the biological phenotypes are changed. So this we mentioned above have been applied to the breeding program of mariculture animals to alter their hypoxia tolerance or other fitness-related phenotypes. Here, I'd like to introduce one case of plasticity induction in abalone. Abalone is an economically important mollusk. The production and harvest of all abalone species represents a $2.7 billion industry globally. However, it has suffered threats from hypoxia events, as we mentioned in the previous slide. And it's a marine gastropod who release eggs and spawn into the ocean through the holes on the shell. Then the fertilized egg develops into a planktonic trophophore and stays at the bottom after developing into a villager. So for this biophysic mollusk, early life history and developmental plasticity are especially important processes because during its early life stages before maturation, abalone is exposed to drastically different environments due to their planktonic status. So based on its economic value and biological characteristics, we use abalone as an animal model and do something about the plasticity induction. So in our study, we exposed the larvae of Pacific abalone, Aleotis discus hainan, to moderate hypoxia from fertilized eggs to trophophores. Then the trophophores gradually develop into juvenile and adult abalones under hypoxia conditions. So finally, we had two groups of abalones. One is the hypoxia group, in which abalones were controlled under the hypoxia condition. Another one is the hypoxia group, in which abalones were treated under the hypoxia condition during early developmental stages and then controlled under the hypoxia condition. We then measured the hypoxia tolerance, survival, and the growth of juvenile and adult abalones as different developmental stages to determine whether early hypoxia exposure induced some phenotypic changes in later life stages. So first, we conducted a hypoxia tolerance assessment for juvenile abalones of two groups in the laboratory. The juvenile abalones were exposed to extreme hypoxia condition, and their survival was continuously observed. According to the results of survival curve analysis, the median lethal duration of hypoxia group is significantly longer than that of hypoxia group. So it was indicated that the juvenile abalones exposed to hypoxia in its early development stages were more hypoxia tolerant than those who hadn't been exposed to hypoxia. Then we transported the juvenile abalones to the sea-based raft farm for 6 months of farming. During this period, there were frequent hypoxia events and persistent high temperature stress during summer. As you can see in this figure, and finally, the hypoxia group had higher survival rates but lower growth rates during summer. So we think the trade-off between stress tolerance and growth might be an effective survival strategy for abalone. Finally, we transported the 2-year-old adult abalones to the land-based farm for the 1 year of farming. During this period, there was high temperature stress in the summer, but no hypoxia event because land-based farm have artificial aeration facilities. As you can see in these figures, the hypoxia group still had greater survival rates and higher growth rates than the normoxia group. This results imply that moderate hypoxia exposure at the earliest developmental stages can potentially enhance the hypoxia tolerance and some other fitness-related traits like growth and survival in abalone. The effects are very, very long-lasting. We are currently conducting transgenerational experiments. We want to find out if the phenotypic differences between the hypoxia and the normoxia groups may be handed down to their offspring, and how does it happen. Our goal is to refine this strategy so that it can be used in abalone breeding programs. Meanwhile, we are exploring the molecular mechanisms that cause phenotypic differences by using some biological technologies. We found that these hypoxia-induced phenotypic consequences were largely explained by epigenetic variations. Epigenetic variations includes non-coding RNA regulation, DNA and RNA methylation, and histone modification. These variations do not change DNA sequence, but alter gene expression, protein translation, and then phenotype. All right, I won't go into too much detail on biological mechanisms. I'd like to talk more about how to reduce the harm of hypoxia in mariculture. First, reduce the pollutants that mariculture itself emits, and reduce the breeding density to improve the welfare of mariculture animals. Second, assess the hypoxia tolerance of mariculture animals and improve the hypoxia tolerance of mariculture species through selection, breeding, hybridization, genetics, selection, and modern molecular breeding technologies. Third, monitor the dissolved oxygen levels and the temperatures in the mariculture areas through satellite remote sensing and buoys. This can help enable documentation of hypoxia events and the establishment of risk warning systems in mariculture. However, such measures have not been widely used. I think more efforts need to be made. Here, I would like to thank my university, SMU, and Mayo, and 2 colleges, and the Marine Basic Biology Lab for their support of my work. Also thank grants from these organizations. Again, thanks for the invitation. Thank you for your listening. So any questions?
All right. Okay. Thank you. Thank you very much, Dr. Yawei-san.
Yeah.
I think you're addressing a very timely issue, and it's a very important to deal with in the maritime aquaculture. We have questions about, one is how many, can you see in the chat box or QE box?
Yes, I can see it.
So when I just read it, the first question, how many abalone generations is hypoxia tolerance capacity is lasting?
Very good questions. Now we are conducting the first generation, so we don't know how many generations, but we want to know how many generations this hypoxia tolerance capacity can last. Yeah.
All right, thank you. The next question is, again, it's a very great talk. Does the hypoxia group have different morphological characteristics?
Yes, it's quite difficult to answer this question, but very interesting question. Now we know that the hypoxia group have a greater growth rate, and it means the shear weights and weights of hypoxia group were larger than the non-hypoxia group. That was we know now. Later, we want to know if the gill or other morphological characters also change.
Okay, I have a question from myself and you. I think it's very good to look at this synergy between temperature and hypoxia, and also in the real world, the natural environment with the maritime agriculture, very often hypoxia has the co-occurrence with the ocean acidification under the eutrophic condition. Yeah, so to your knowledge, is there any similar studies to look at the synergy between temperature, hypoxia, and ocean acidification?
Yeah.
I know it's a
I know that the high temperature and the hypoxia co-occurred very often, but I don't know the ocean acidification, and the combination of these three factors, I think, is very important important in biological experiments and also in biogeochemistry. I think the combination of the two parts is very important.
All right, okay. We have another interesting question. Does the hypoxia tolerance group taste the same as the normoxia group?
Very good question. If you want, you can comment taste our abalone to compare these two groups. Welcome, very good, welcome.
Yeah, I assume it's an ongoing work, so it's a good topic to check in. And it's just so Jeremy Steering from the Secretary IOC UNESCO notice. Please check back if we will have some online answers to the questions you posted. Okay, we have another question. Again, nice and interesting talk. Is there any effort to lower the density of marine culture or move cage to lower the impact of the sediments on the sediment, thus reduce hypoxia in the user environment?
Lower the impact of sediments.
So I think the context was that when you have these enrich the feeding process, many of the organic material will be settling down in the sediment. In the long term, that in fact
okay, I know that
the environment. Yeah.
First, I think from this scientist point, it's necessary to lower the density of marine culture or to move cages. But I think this is not that easy to do this because we know that economic efficiency is very important to businessmen and the farms. So for researchers appreciate collecting data and finding solutions to scientific problems. The operation is very difficult. So future works will focus on bringing together stakeholders to address the issues of hypoxia, also to other problems, for example, heat wave, acidification, and pollution in mariculture. The cooperation of these stakeholders is challenging and important to, like the Christopher said in his question. It's easy to see lower the density of mariculture animals and lower the impact on sediments. And but who will do this? Right.
So there's a cost issue and in this marine content, the real business, I would say, yeah. Is that right?
Yeah. Thank
you. All right. Thank you again very much, Yawei. And can you unshare the screen and we'll move to the next speaker.
Okay, thank you. Thank you very much. Sorry.
Click on the top of the screen.
I found it.
Yeah.
Thank you.
So let's welcome the second speaker, Folco Giomi. Giomi, floor is yours.
Thank you very much. And thank you very much, Minhan, for the moderating this event, and of course to the committee for organizing this wonderful the wonderful meeting. Today I will not stay close to the topic of the oxygenation, but I will talk more generally on the subject on how marine life perceive the oxygen availability, or great part of marine life perceive the oxygen availability in the ocean. And indeed, as human being terrestrial animal, are prone to read the oxygen availability as an homogeneous systems that tend to the ideal standard of saturation, so the 100% of saturation. Consequently, any small or large deviation from this condition is generally seen as a possible threat for marine life. Today, a large scale and a long-term monitoring of oxygen in the ocean has revealed a progressive decline of the global oxygen content, and this is surely a potential threat for and an important issue to address. But it appears loosely informative of how the great majority of marine life perceive oxygen availability and the consequent ecological relevance. Thus, trying to focus on the significance of oxygen availability, really significant for marine life. Today, I'll try to show you a couple of examples of, in particular, the example of three major players, which indeed are just the representative of three different cohort of organisms, and how this example rule, cope, and get benefit from the great fluctuation of oxygen in marine systems. I will start with the primary producers. So indeed here is represented by the phanerogam, but many other marine primary producers are represented by this example, and ranging from very tiny and well-distributed algal or bacterial mats to brown algae, phanerogams, and any other kind of marine primary producers. And these are data we have collected in the field, and as you can see, there is an amazing variation of of the oxygen in the field. This data show the parallel fluctuation of oxygen and temperature in a productive habitat. But what is impressive is the variation of oxygen that can range from a very low value close to the hypoxia or even anoxia to impressive massive production of oxygen in condition of up to the condition of supersaturation, up to 250% of saturation. These are more data that comes from the Venetian Lagoon, and it's a long-term data systems show that show a sort of, when average, a sort of linear trend around the value of saturation. But when we close up, so when we have a much more detailed analysis, it emerge a great temporal heterogeneity, and this is the variation of oxygen on a daily basis or even on every 6-hour basis. And as you can see, there is a much more complicated scenario. Underlying the importance of adopting a great temporal scale to really get the temporal heterogeneity in oxygen availability. If we close up on a few days pattern, you can see with the red line a sort of average trend close to the under saturation value, around 80–100% of saturation. But the daily pattern at 1 minute scale is much more interesting and much more relevant, and emerge a pattern that it's really affect marine life. This is the pattern where marine life has to cope with. But we can even get more interesting, another interesting feature if we move to analyze the spatial heterogeneity, so moving at the geographical level, and the concept of spatial heterogeneity become relevant, showing an impressive range of fluctuation, where you can see on the vertical axis the great variation, but what is particularly relevant is the site-to-site difference. So there is a sort of site-specific fluctuation range of oxygen in the field. This underlines the importance of adopting a site-to-site analysis of oxygen when we want to describe a certain pattern experienced by a community. Another important aspect is that emerge is the data above the 100% of saturation. So the occurrence of supersaturation, that is again vary site per site, but show the condition of oxygen enrichment above the saturation, and that occur daily. So you can see how great variability of oxygen saturation in the field is quite consistent. This oxygen saturation is a generally neglected aspect in the literature, because we mostly focus on the undersaturation, so hypoxia and anoxia, while oxygen saturation is a common environmental condition that occur daily, and is a really fundamental feature of productive ecosystem. So here we have data from different environment. We have collected from North Sea to the temperature in the Venetian Lagoon, or in different environment in the Red Sea. And finally, just to show you other aspect of the variation of oxygen and in the field, so the fluctuation of oxygen in the field, you can see on the right, and on the right corner of the a plot showing a cross wavelet analysis between temperature variation and oxygen fluctuation. And as you can see, there is a clear in phase covariation of temperature and oxygen. So these parameters are perfectly in phase with a 1-day period. That means that each temperature, each oxygen supersaturation peaks when the temperature peaks. And this is very important for marine life, and we will talk more later. But even more clear is the left panel, where you can find a 1 degree bean plot. So each box plot represents the oxygen variation at every 1 degree bean. And as you can see, all high temperature beans are characterized by only supersaturation. That means that in the field, when high temperature occurs, there is always and only supersaturation, no undersaturation in such productive habitats. Now I move to the second player. So I will take an example of a sessile organism. This case is quite a special case of a sessile organism because I took a coral. This is special because it's not only consume but also produce oxygen thanks to the symbionts. But most important, it will pop up the response of oxygen fluctuation. But more important, it's quite fundamental today because it's one of the coral reef are one of the greatest example of one of the greatest topic for the study of oxygen, of the oxygenation in a marine habitat. Indeed, the large body of studies have investigated the occurrence of ocean deoxygenation and the effect on coral reef, underlining a certain high effect, a higher vulnerability of coral to hypoxia. So it seems that corals are really vulnerable to hypoxia, but both on the sort of spatial scale, but also on the temporal scale. These are data along 24 hours. And as you can see, coral seems to be more vulnerable to hypoxia than other organisms that inhabit the same environment. But the question is really the environmental oxygen we measure and the average of environmental oxygen really meaningful for this species, for this ecosystem engineer? Is the link between the average environmental oxygen really relevant? And trying to answer, I want to show you some data recently published. It's very interesting studies, and it's a great example that show the real oxygen availability perceived by the coral, and it emerged that everything the coral experience happen in the close nearby of the compartment, happen in the boundary layer around the organism. The boundary layer is the tiny area enclosed below the gray dashed line, and you can see an amazing variation of oxygen, even magnified if you move from the darkness to light. So when night to day, when during the day there is a massive production of oxygen by the symbionts, showing that the close surrounding of the coral is really hyperoxic, experiencing a clear oxygen supersaturation. And this is what really the coral experience, not just the average surrounding the coral reef area. This is even more, this has been already suggested by data in the early 90s, showing that the importance of this boundary layer is generally underestimated, because there is a great and extreme fluctuation of oxygen surrounding in this case, the polyps. So this founder organism. These are the pattern during the night, where you can see a dramatic decrease of oxygen around the living organism. But during the day, this maximizes, so the oxygen really increase to incredible high value, like close to the 200% of saturation, and the coral really experience a condition that usually are generally neglected when we average the data from the environment. And moving to the effect of oxygen supersaturation and thus moving to the effect of such daily fluctuation of oxygen, I will move to the third player where I took a opportunity crabs, and in this case, Talamita crenata, which is a great example for benthic or marine organism that inhabit such highly fluctuating environment. We tested the Talamita crenata in a temperature gradient, so in increasing temperature, a classic increasing temperature experiment, and we measured the mortality. This is the mortality curve at 100% of saturation. But what is astonishing is that when we measure the oxygen, the temperature tolerance at under 40% of saturation, we recorded a clear right-hand shift of the curve. So the organism acquire a great tolerance to temperatures of up to 4 degrees. So it becomes more tolerant to increasing temperature. And this is quite a fundamental because, as you remember, I will show you that high temperature occurs only when there is high oxygen. So the two are synchronous and parallel. So this organism experience extreme heat, but also experience the very high supersaturation that provide a much more interesting resistance to temperature. This is not just restricted to this organism because we tested the same experiment in 18 species from 6 different phyla at 3 latitude from North Sea to Red Sea. And this is quite a constant result. So all the organisms we have tested are really prone and really resistant to temperature when exposed to the daily occurrent supersaturation. Which is the mechanism behind this effect, which is the mechanistic background of these results. And in our opinion, it stands on the theory of the oxygen capacity limitation or thermal tolerance, where that postulate that the thermal tolerance is driven by the oxygen availability. So high oxygen availability can boost the thermal tolerance, while a mismatch between oxygen demand and oxygen supply can impair the thermal tolerance of organisms. So we tested on Talamita crenata this effect again, and we measured the oxygen consumption, so the respiration rate, the metabolism of Talamita crenata in again in a temperature gradient, and this is the pattern. At 100% of saturation, as you can see, there is a clear exponential increase of metabolism up to 38 degrees centigrade, but and sudden a decrease of the aerobic performance. So 38 degree is the critical temperature for this species. But when we measure the same oxygen consumption rate and under 40% of saturation, a quite different pattern emerge. So there is an extension of the aerobic performance in a supersaturation. So the organism is able to reach much higher metabolic rate, much higher response, but most important, can increase and sustain its metabolic performance up to 2 degrees further. So become more tolerant and more active. This is not the thermal tolerance, the gain of thermal tolerance is not the solely effect that oxygen supersaturation can provide. Indeed, we tested other effects, and we showed that the exposure of oxygen supersaturation provide an impressive tolerance to hypoxic event. So experiencing hyperoxia during the day can mitigate the effect of hypoxia or recover the effect. Specifically, oxygen supersaturation can decrease the critical PO2 of an organism, so it can withstand a much severe hypoxia, or can increase and maximize the efficiency of lactate recovery after an hypoxic event. And this is quite important because, as you remember, the data on the coral that demonstrate the tolerance of, or the vulnerability of tolerance to hypoxia, possibly we interpret that measuring at the right oxygen concentration, the one that's surrounding in the boundary layer the polyp, it can provide a more reliable and possibly, hopefully, a more robust tolerance to hypoxia than when it's measured at an average condition. Just to conclude, I want to push people to try to include supersaturation in their experiment, because it can provide a much more interesting and robust results. And then I want to suggest that I want to underline that the rate of biological process measured at an average of oxygenation does not match the average of the same process under realistic oxygen variability. Do include variability in your study. And of course, conservation is the priority. Thank you very much for your attention, and I'm more than happy to receive questions.
Thank you. That's what's wonderful. Yeah, thank you very much for your excellent presentation. And before I move to the question online, and I do have a question about this. So you emphasize the temporal variability in addition to the main stage of this the oxygen level and their impact on the marine lives. That's the essential message you want to deliver. So what about if you look into the evolution point of view, what's this type of the effect would be examined?
Indeed, it's quite curious because almost all these organisms evolved in such a highly fluctuating environment, because coastal environment or other marine productive environment like the photic zone in open oceans are greatly fluctuating environments. So on the evolutionary time scale, marine communities evolved under a fluctuating environment. It's us as scientists that start to include the fluctuation only in recent years. And in my opinion, the point is what all the literature and the data we produced in all our scientific decades under very stable and steady condition in laboratory with 100% of saturation, what can tell us? Can we really say that, I don't know, just to make example, an experiment on ecotoxicology made at 100% of saturation in laboratory, is it really ecologically relevant? Or we can withdraw a different result if we include the real fluctuation of oxygen that organisms should experience in the field.
Okay, that was a follow-up question. So lead to the amplitude, that's the variability, and also the frequency of this low and high oxygen saturation. Which factor are the most important one on the tolerance or resilience of the marine life?
In my opinion, higher the fluctuation, higher is the effect. Of course, the fluctuation can be negative or positive, so down to the hypoxia or up to the superoxia. But in my opinion, the magnitude of the fluctuation, which is generally coupled with the magnitude of the temperature fluctuation, is the pivotal element to look at. And another important aspect we are trying to investigate right now with my colleagues is the predictability of such fluctuation. So how the organism can predict, in a certain sense, the weather of tomorrow, so the fluctuation of tomorrow. And in our opinion, this predictability feature can be a great driver of the evolution of marine organisms, diverging species, organisms between specialists and generalists. Like specialists are those with a higher predictable capacity and generalists with a lower predictable capacity.
Thank you.
You're welcome.
Folco. We have questions from the QA box. And Yawei was asking why you choose 140% in saturation. And I think it's quite high in saturation. It's in very high productive area you'll find that. Is that right?
Sorry, I didn't get your point.
So why you choose 140?
Okay, yeah. I took
saturation. You do the experiment.
You agree. It's a general. We took a sort of average value because when we did this experiment in Red Sea, we collected data ranging from saturation up to 180% of saturation. So we took a sort of average value in this super saturation range. But you're right. It's if we did at 130, 150, we can get slightly different results. But we should try to we have to demonstrate a pattern. So we choose a sort of mixed high value.
Okay. Thank you. Christophe Ribouille has this question. Could you expand on the synchrony between temperature and oxygen, because oxygen saturation decreases with temperature? How is the synchrony achieved then? Is it only in shallow environment?
This is a good point, sort of old-fashioned ecology point, but I agree. I should suggest to read the article made on ecology by Wilco Verberg on the oxygen availability index. But to answer this question, is it true that oxygen saturation decreases with temperature? But at this amazing production of oxygen determined by the primary producer, the effect of temperature on saturation is really not, I do not say neglectable, but really low meaningful, because the production of oxygen is so massive during the day that really overtake the effect of saturation. Is so massive that you can get oxygen, bubble of oxygen on the substrate. If you, I don't know, if you pass your hand on the algae or on the mat in the coastal, when you grab on your coast, you see a lot of tiny bubbles just blow up. These are pure oxygen, and so the synchronicity is always there, because temperature boost, sorry, the solar activity during the day boosts the temperature, and the solar activity boosts the primary production. So the two are parallel and occur almost exactly in the same time. It is only in the shallow environment? No, it is only in a productive environment. So in the sunlit zone, for example, in the open ocean, the first 150 meters, 100 meters of depth, where there is a huge amount of algae, the synchronicity is there and the oxygen production is there.
Okay, thank you, Folco. So this next question is, how do different players interact in the warming environment affect the ecosystem as a whole? And I think the question was tried to the holistic view. Yeah.
This is a complicated question because it requires almost an entire ecology course. But in my opinion, all organisms get synchronous with the consumption and production of oxygen in the field. During the night, they are all consumers, so all metabolism try to cope with the progressive decline of oxygen. But during the day, the primary producer become productive, so boost the oxygen in the field, and all other compartments of the community.
Okay. So the next question is to ask you to refine the take-home message with respect to the deoxygenation issue.
The players is fundamental to really describe the connection and the stability of the ecosystem at work.
Okay. Okay. So the next question is about the take-home message in relation to this deoxygenation issue. So I think the question was asked you how the effect we will see in the low oxygen condition.
Yeah, the question seems like I'm a bit off topic in this meeting. I agree. Indeed, I'm not really talking about the oxygenation at all. Not in the classical sense today. So the progressive decline of oxygen in the ocean. I'm more interested in the daily deoxygenation, a certain ecosystem experience, like during the night, because during the night, marine ecosystem, productive ecosystem, almost always experience a progressive deoxygenation, which is almost totally recovered during the day. So if you want a take-home message regarding the oxygenation, my suggestion is take a look to the resistance of organisms to short-term deoxygenation, like a few hours during the night, and how they recover during the day. Possibly we will understand something better about long-term resistance or resilience to the oxygenation. So taking a look to what happened daily, every day, will provide some interesting hints on what we should foresee in the future.
Yeah, I think I would like to echo Folco that the fluctuation and the variability in the short-term time scale is very important to look into this real ecotoxicology of the deoxygenation. Even that's I understand. I think we'll touch on one more question. What is the physiological mechanism for the decreased mortality in supersaturated oxygen condition?
Okay. Because supersaturation can mitigate the effect of temperature. The thermal tolerance induces the capacity to withstand not just the critical temperature, but also lethal temperature, is driven by the availability of oxygen in the body compartment. So if an animal is able to extract enough oxygen, can tolerate higher temperature. This can be provided by increasing the ventilation of the cardiac circulation of the systems, but in a supersaturation environment, the capability to extract oxygen from the water is maximized. So the organism can really take benefit of the such hyperbaric condition like those of the athlete for human, and perform better, and thus mitigate the harmful effect of temperature and consequently be less prone to die at the same temperature, increase the survival.
Okay. Thank you, Folco. Thank you, Yawei.
Thank you very much, Minhan.
To both of you for the excellent talks, and also to all the audience for your participation to this webinar. And we very much hope to see you all again on our next webinar in February. And be sure to follow us with any of the social medias and in the meantime. And please feel free to find more information there and sign up for our current call for information and our activity for World Ocean Day in June this year. So thank you all very much again, and I'd like to close the webinar today. And thank you all. Thank you.